information contained in this video and/or audio is provided as is for educational and informational purposes only and should not serve as the basis for any trading or investing decisions. Little Grapevine makes no representations and disclaims all express, implied, and statutory warranties of any kind to any viewer or other third party. Neither Little Grapevine nor any of its affiliates makes any endorsement of any particular company, security, product, or financial strategy, and nothing contained in this video and/or audio should be construed as investment advice. Please be aware that we and participants viewing presentations and/or live webcasts and engaging in Q&As may own, buy, or sell shares of featured companies. Investors should undertake their own due diligence and carefully evaluate companies before investing. Please see safe harbor statements of companies participating in Little Grapevine events by viewing their videos and slide presentations at littlegrapevine.com.
I really believe that the CEOs talk way too much. I'm the CEO, chairman, and founder. I built a biotech company called Iovance from zero to almost $7 billion. There's no way I can give justice in 90 seconds to what the trike really is, but all I can say is that the next wave of technology isn't cell therapy, which I launched, the first cell therapy company. It is targeted immunotherapy, particularly using NK cells. The trike's data through patient 9 has shown in a phase 1 of a phase 1/2 clinical trial, where in phase 1 you're just supposed to be able to establish maximum tolerated dose and safety level. We're seeing efficacy. The last 5 patients, 3 out of the last 5 patients, once we got to 25 micrograms per kilogram, up through 9, patient 9 at 100, have shown complete responses or remissions in phase 1. You never see that data. If that doesn't get your interest, you're not into biotech. And biotech data, human data, that's what has to that's what you should listen to. Because clinical trial FDA data, you can't lie about it. You can't paint it in anything other than what it is. It either works or it doesn't work. And then our clinical trial data is what has caused us now, since the ASH conference on December 5th, where we were still a bulletin board company a little over a month ago, it caused us to be able to uplist to NASDAQ, to raise about $54 million to clean up the balance sheet, to have 33 million shares fully diluted with no encumbrances. And it did it because our data, when we presented it at the ASH conference, which is a 30,000 member oncologist, hematologist, biotech conference, hundreds of companies present there. They put their posters up, their abstracts, and out of that group, the ASH conference coordinators picked 5 companies to do an oral presentation, only 5. There was Merck, Celgene, Pfizer, Novartis, and number 5 was us as a bulletin board company. They'd never done that before. Why? Because the data that we showed up through patient 7 all of a sudden said, these guys have something real. So they asked us to do the oral presentation. That allowed us to get a lot of interest. And that allowed us to complete our NASDAQ uplisting, raise all that capital. I have enough cash for 2 years now to execute, don't need money. And that was really what happened to us. Now the presentation part of it, I'm going to focus more on the data because I actually like to practice what I preach. All right. And that is to tell you why you should listen and why just forget about the CEO speak. All right. Let's tell you where the company's really at. The other thing I like to do is I hate CEOs that sit there and tell you what they're going to do. Here's what we're going to do in the future. This is what it looks like. So I think that's all bullshit. Because I like rather talking about here's what we've accomplished, here's where we're at, and the people that we speak to are pretty smart folks. You guys can connect, our gals. You can connect the dots. You can kind of figure this out. So I'll give you the basics of what the trike is and what its data is saying, and that's really what you need to focus on. 1 minute on Iovance. I created Iovance, which became a very large company, which is a cell therapy company. In our landscape right now, what I felt when I left Iovance, everyone thought I was going to create another cell therapy company. But what I liked about cell therapy and what I hated about it was I like it because on stage 4 patients that have nothing left to do, cell therapy does have efficacy and it has saved some lives and it will continue to do so. But it's a very limited audience. You can only service 4,000 or 5,000 patients a year because it's $1 million to $2 million per patient. So its economic model is really severe. There's not a lot of margin in that either. Then the ergonomic issues with cell therapy because it's not really a drug. It's a cell therapeutic regimen, which you have to do a lot of conditioning of patients, a lot of genetic engineering, re-engineering, and NK manufacturing, a number of things, which causes this massive cost. And the ergonomics of it, the patient experience is a lot of side effects. It's really tough on patients. And the physician experience is tough to administer. And it's also tough to find insurance companies that will cover $1 million to $2 million per patient. When I left Iovance, I went, look, conceptually, I like CAR T. I had that asset for a year before I had to give it up because I was a small bulletin board company and I couldn't afford to do both companies. Iovance did fine. The CAR T I gave up ended up with Kite Pharma. They sold it for $12 billion to Gilead. I'm never giving stuff away again. That being said, CAR T is still a cell therapy. It's limited to liquid tumors. The Trike technology focuses on where I felt this was all going, targeted immunotherapy. NK cells are really, really prolific cancer killers in your immune system. T cells are what everybody kind of understands. There's a lot of product out there with T cells, but T cells are like soldiers. Your immune system assigns T cells to kill a foreign invader, and they go in mass. And T cells, for a while there, they do their job, but then they become a problem because they actually can transition to something called a cytokine, which becomes very dangerous. Because like a soldier in the field, once it gets distressed or overused, it becomes a problem. NK cells are the most prolific cancer-killing agents that the immune system creates. They are the biggest cancer-killing biology in history. Everybody has NK cells, but by the time you get to stage 4, which is where most cancer treatments get approved in stage 4 patients that are terminally ill, your NK system has had the crap kicked out of it. So you can't, NK cells have a problem in that they only last about 3 hours, so they don't last long. So you're constantly having to replenish NK cells. And in a very sick person, your immune system's not that efficient anymore. Also, in the landscape that's out there with NK cell engagers, which is what we are, there's Affimed, Dragonfly, Innate Pharma. Those are NK cell bispecific engagers. The lowest market cap in the NK cell space is Affimed, at about $900 million market cap. And then you have the NK cell therapies, which are again a cell therapy that sells for massive amounts of money. And that's Nkarta, Cellularity, Fate Therapeutics, which is at the top of the food chain at about $10 billion market cap. And oh, by the way, that NK cell therapy was designed and developed by Dr. Jeff Miller and Dr. Rhines out of the University of Minnesota. It's their older technology. It's working well. Cytovance has huge valuations on it, but it's still a cell therapy with all the economic side effects. They're also, it's not a monotherapy. Even they use something called Rituxan as a combination drug therapy because in and of itself, it doesn't really complete the job. All of the NK technology is trying to do the same thing. They're trying to take an NK cell, target directed to a cancer cell and kill that cancer cell. And to some degree, all of them have a modicum of success. They can get an NK cell to target a cancer cell, and then that NK cell, because it doesn't last beyond 3 hours, or it becomes exhausted as soon as it does it, and it doesn't work anymore. So that's why all of those NK technologies, whether it's the NK engagers or the NK cell therapies, all need supplemental assistance to help their drug work. They all need outside NK manufacturing because the body can't create enough NK cells. That's really super expensive. It also isn't biologically as good as an NK cell that your immune system creates because it has a thing that it suffers from called clipping or shedding of CD16, which is the target. The Trike is different. The Trike does everything in your body biologically. With the Trike construct, so the Trike, instead of an NK cell killing just one cancer cell, the Trike's a serial killer. The Trike platform, it's a platform technology. So the targeting part of it is an NK cell with CD16. All NK cells have CD16 attached to it. That's the target. IL-15 is our secret sauce. Interleukin 15 is a known cytokine. You can only administer IL-15 at 2 micrograms per kilogram. Anything above that becomes very, very dangerous. We're at, on patient 9, 100 micrograms per kilogram with not one side effect in the patient on anything. It really was a concern of the FDA's initially because we incorporated IL-15 in the middle of our small protein biologic. For those of you who don't understand the dynamics of biology in your immune system, a molecule, if a molecule were the size of the planet Earth, a small protein biologic is the size of a satellite. It's very, very small. Its DNA is extremely sophisticated. That's what the trike is. The trike is the protein version of CAR T. But instead of CAR T only working in liquid tumors and being a cell therapy, it is a drug. It works in liquid tumors, solid tumors, and infectious disease. It's economics because it's a small protein biologic. Instead of $300,000 to $500,000 per patient cost, it's $30,000 a patient. Everything the trike does is inside the body. No outside NK manufacturing, no genetic engineering, no preconditioning of a patient's immune system. None of that. It all happens inside your body, making it different. And what really separates the trike is, remember I told you that NK cells have a very short life cycle, 3 hours. This is actual human data that you're looking at. This is a video that I'm about to play in real time. This is a microwell from a special MRI video photography. That little purple thing in the middle there is an NK cell then the trike paints that NK cell and target directs it. Those radioiodized green things are leukemia cells inside of a patient's body. You'll see the NK cell here. Remember I said all the other technologies, the NK cell dies as soon as it kills a cancer cell. It's exhausted. It doesn't work anymore. You'll see that the IL-15 component of our trike and the way we put it in our construct allows us to deliver. IL-15 has a very symbiotic relationship to causing NK cells to excite, to expand, to persist. Everybody out there tries to use IL-15, but they can only use it systematically at 2 micrograms per kilogram. Our patents allow us to take, and we're up to 100 micrograms per kilogram because an NK cell responds very well, causing, when our IL-15 attaches to that NK cell, it causes it to do 3 things. 1, to proliferate, create more NK cells with IL-15 inside the body, not outside the body like everybody else. 2, it causes that NK cell to persist, all right, to stay for 3 months instead of 3 hours, which is its normal life cycle. And 3, it causes it to become a serial killer. So what you're going to see right now is in real time. This is not fake video photography. This is real human data in one of our patients. You're going to see that NK cell acting like Pac-Man. Those leukemia cells, it's looking for CD33 positive. That's our target for that first trial that we're in. We're almost finished with phase 1 now, getting ready to go to phase 2. You're going to see it kill a cancer cell and then continue to kill. And this is it doing its work. It's looking for CD33 leukemia cells, which is all present in all those liquid tumors. When it turns red, it's killed those cancer cells. And it didn't kill itself. It's still working. And it's still going to look for CD33 positive leukemia cells, but it's not going to kill anything other than the cancer cell. That's why we're not showing any side effects, not even fever in these patients. And now it's killed 3 of those cells in the microwell, and it will do that for months. And it will do that in billions of NK cells because it causes NK cells to proliferate. That is real data. Now let's get into the clinical trial part of it, because that's really what matters. Right now, the trike works in multiple mediums. Like I said, liquid tumors, solid tumors. Liquid tumors are 20% of the cancer market, solid tumors are 80%, and infectious disease. We're almost done with our phase 1 portion of the trike. We just started treating patient number 10 on Monday, and that patient will be finished in 3 weeks. There's 3 4-day cycles. We'll have 2 more patients. That's at 100 micrograms per kilogram. We'll have 2 more patients after that, 11 and 12, and that will complete our phase 1 study. The FDA has already approved us to go into phase 2. We'll begin our phase 2 study in June. And that's where we're at. Now, the other trikes that are on this list are solid tumor trikes, because the trike works in solid tumors. It's not limited to just liquid tumors, which is 80% of the market is solid tumors. We have a PDL1 trike. That PDL1 trike, we can target that for colorectal cancer. We have a B7H3 trike, which is in manufacturing right now. That hits multiple cancers. And so that hits pancreatic, prostate, and ovarian cancer. So it's a basket trial. And then we have two other trikes, which are in the same level right now, being in manufacturing. One called HER2 for breast cancer, and another one called ROR1, which covers metastasis, stopping metastasis on cancers. Another one. We have 22 different trikes. So where that leaves us, let's talk about the data, which is the thing that I keep telling you is the only thing you should listen to. The trike is administered in 3 4-day cycles. It's 4 days on the drug, 3-day break, 4 days on, 3-day break, and then 4 days on, and you're done. When we bring these patients in, we measure two things. Their NK cell levels. These are stage 4 patients ready to die. They failed everything. So we measure their NK cell levels, and we measure their bone blast levels, which is the amount of leukemia or MDS, myelodysplastic syndrome, cancer cells that you have in your bone marrow. That's the percentage. That's what you're really infected with in cancer. That being said, because this is a first-in-class, first-in-human drug, this has never been done biologically before, and because of our use of IL-15, the FDA was very concerned. They were worried about side effects because IL-15 used over 2 micrograms per kilogram is deadly. So they said, look, when we did the animal models, what they want to see is at what level of dose can you get up to, and then when are you going to start killing the animals? They want to see the animals die. Because they want to be able to know that what toxic level there is if you're taking and putting this drug into humans. At 1,000 micrograms per kilogram, we couldn't kill any of the rabbits or mice, but we were killing all the cancer. And the FDA went, when is this thing going to tox out? We went, it doesn't work that way. So they finally got their hands around that and went, okay, it appears that you're doing things safely, but we're concerned about IL-15. So the first 2 patients, we're going to make you very low dose that patient at 5 micrograms per kilogram. That's that lower box in the left-hand corner. Then they said, if that works, then we'll let you go to the next 2 patients at 10 micrograms per kilogram. And if you still have no problems, we'll let you go to the next cohort at 25 micrograms per kilogram. No problems there, 50. No problems there, 100, no problems there, 200, completing the 12 patients in the phase 1 portion. So patients 1 through 4, all right, even on the first patient at 5 micrograms per kilogram. Well, remember, we measure their NK cell levels and their bone marrow blast levels. That patient's bone marrow blast levels was increasing. It was at between 5 and 10%, but it was increasing by 10% every day. That's why that person had 6 weeks to 2 months to live. So we had to tell the first patient, it's not likely that anything is going to be as effective for you because 5 micrograms per kilogram is a very low dose. So that patient came in very sick and needed assistance in a wheelchair. After the first 4 days, we started seeing energy coming back to that patient. They were able to walk again, didn't need the wheelchair anymore. So we knew something was happening. When we finished, we can't measure their levels until after they finished the third 4-day course of the drug. And then 10 days, we have to wait 10 days, and then we can test their bone marrow levels and their NK cell levels. What we found in patient number 1, right out of the gate, was that her NK cell levels went back to normal. That's where her energy level went up and her appetite and everything else. And that her leukemia levels stabilized. They didn't increase anymore. That is a win. Phase 1, you don't see signaling ever. It's rare. I won't say ever. It's very rare. And then the second patient had the same impact. NK cell levels. Patient number 1's NK cell levels was at 0.003%, increasing to normal after the 4-day course of the trike drug at about 8%. Same thing happened to patient number 2 at 5 micrograms. Stable disease. NK cell levels went back to normal. That's another win. When we got to 10 micrograms per kilogram, same thing happened. Patient number 3, stable disease, blast levels still didn't have much impact, energy levels back up, still no side effects, not even a fever. These patients all have major fevers. They have cardiac events, pulmonary issues. Leukemia patients and MDS patients are really, really sick people. When we got to after 4, the FDA said, okay, still not one side effect, not even a fever, keep going. And now we're delivering IL-15 at more than twice its normal dosage without any side effects, because IL-15 always has side effects, but not with the trike. When we got to patient number 5 at 25 micrograms per kilogram, because we were still showing safety, that patient's blast level, that's the amount of leukemia in his bone marrow, went down from 12% down to went from 18% down to 12%. Now it's showing killing of the leukemia load. It's killing the cancer cells. That's unusual in a phase 1 trial. And 25 micrograms per kilogram is still a very low dose. Patient number 6, NK cell levels went back to normal, didn't have any impact. Patient number 7, when FDA approved us to go to 50 micrograms per kilogram. And that patient, she was an MDS patient. Nothing works for MDS, right? It's a really nasty disease. But her bone marrow blast levels of MDS cancer went down from 12% to 4.6%. Anything under 5% is a remission, a complete response. So we're seeing efficacy in phase 1 where you don't see that kind of thing until you get to phase 2, hopefully. So now we're shown in patient number 5, we reduced the cancer level down by 33.3% in leukemia. Patient number 7, we reduced the cancer MDS rate by 61.7%, killing the cancer in the bone marrow blast. That's a huge win. Anything under 5% is big. That's a CR. 50 microgram at the next 50 micrograms on patient 8, really no effect, right? For whatever reason, that patient also had liver disease. We think that impacted this. When we got to patient 9 went down. Their bone marrow blast went from 22% to 8%, another complete response. That's huge. You're seeing efficacy in phase 1 where you just never see efficacy until phase 2. Rarely, actually, I shouldn't say never. It's extremely rare. So once we got to where it started showing cancer killing at patient 5, 3 out of the next 5 patients got a complete response. Those are approvable numbers by the FDA. That is big time data. Patient 9 cancer reduction level went down by 63.6%, increasing at every cohort that we increased the dose. Our next 2 patient 10 started on Monday. We expect similar results on 10, maybe better. Patients 11 and 12 are in the queue. Once we finish that, we'll start our phase 2. We'll hit another big milestone to start our phase 2 trial. So that is the data that I'm talking about, minus all the BS from a CEO. So now I'll open it for questions, and happy to do so. Well, the competitors get an NK cell and they find a way to target direct it using sometimes genetic engineering, sometimes preconditioning of the immune system. There's a lot of outside forces going on in there. And when they do that, and whether it's NK cell therapy like Fate or NK bispecific technologies like Affimed, Dragonfly, Innate, so they can get an NK cell to kill a cancer cell, but when it kills that cancer cell, it also is spent. It can't kill anymore. So they have to have another NK cell because of its life cycle. And once it's done and it kills a cancer cell, it's no longer useful. Because of the incorporation of IL-15 in our NK cell technology, causes that NK cell to kill a cancer cell, but it doesn't die. It becomes a serial killer. It stays along because of our IL-15 component. It causes that NK cell to continuously kill other cancer cells, not just killing on a one-to-one relationship. It can kill hundreds, millions, billions. It lasts for 3 months now instead of 3 hours. That's what the trike does that nobody else can do, because the trike doesn't have an issue on the CDC targeting part of the NK cell called clipping or shedding, which all of them do. When you do an outside manufactured NK cell, it doesn't incorporate this thing called ADAM7, which connects the CD16 to the NK cell, allowing it to persist. The trike does it all biologically without any assistance, and it's using your immune system's intelligence to leverage and continue to not only keep an NK cell alive for months, but also to cause that NK cell to now proliferate and continue to in mass. So it's an exponential event because of the trike. In the phase 1 trial, the first phase 1 is for leukemia, acute myeloid leukemia, and MDS, myelodysplastic syndrome. We have 22 trikes, right? 4 of them are being in manufacturing right now for the solid tumor trikes. I have enough money to finish my trials and take these other 3 solid tumor trikes. I have enough money for 2 years right now. I don't need money. But that being said, the best time to raise money as a biotech company is when you don't need money. Okay? Today I was offered $100 million. Danny was in the car with me by a bank that said they would underwrite it, but I don't want to raise money right now. And I'll raise more money to bring other trikes into it, but I'll do it in September when I think our market cap value might be as close to $1 billion. It probably can hit that. Because if you look at the landscape, we're the lowest guy on the totem pole at 260 million, because we were a bulletin board company a month ago, and we're no longer that. So I think we're scaring the hell out of the competition. I'm happy with that. I'm ready to have that fist fight with them based on data and achievement and milestones. All of those NK engagers, they're bispecific. They're not trispecific like the trike. They don't have multiple targets like we do. They all have big pharma partners. It's not unrealistic to think we will too. Before I put a trike in the manufacturing pipeline, we have data, right? We do all the animal models, the enabling studies, the tox studies, and everything else. So I already know that my trike's going to work before I manufacture it. Otherwise, why go into that process? Because then you're just throwing money out the door. So the things that can go wrong with the company right now are not company specific. They're really sector specific. If the biotech sector has a problem because of Biden politics or, a World Trade Center thing, we can't control that. And that would hit the whole market, all right? And we have a clean balance sheet, and now we're NASDAQ. I have an analyst report from Roth Capital that came out a few weeks ago at $25 price target. I have bulge bracket banks that want to give me money right now. I don't want money. I won't say I won't take money, but only at higher valuations, right? I'm not going to dilute myself and all the people that have come in, right? We don't need to. We don't need the cash. But you take the cash to build the product pipeline up. I have Big Pharma. One actually asked me if I'd sell the company now. I told him the answer is yes and no. So I want to kick that can down the road. I'll have one partnership with Big Pharma. I could partner out all 22 trikes, but that would just dilute the value of the platform. I'm not going to do that. I want one Big Pharma with 1 or 2 trike candidates. That'll give us credibility and non-dilutive capital in a big way. Affimed got 96 million up front from Hoffman LaRoche. Dragonfly got $750 million from Merck. Our technology's outperformed. There's a company called Nkarta, an NK cell therapy. Market cap's about $1.7 to $2 billion. They treated 1 patient in November with 0 results. And when I was doing my NASDAQ raise, I brought that up, and the guy said, well, Tony, they got pedigree. I'm like, that's bullshit. Not more than us. The number 1 NK specialist in the world, the most published person whose previous technology is at Fate Pharmaceuticals as an NK cell engager at $10 billion market cap, is Dr. Jeff Miller. The most published, the most knowledgeable, all NK cell technologies go through Dr. Miller, all of them. Every banker, every analyst checks with Dr. Miller. And he goes, well, that's true. Jeff Miller's number 1 guy. Okay, I said, okay, now let's look at their management. It's a doctor from Columbia University, medical school. Never run a NASDAQ company. Never created one. I created one worth $7 billion. So who's got better pedigree? He went, well, okay, you guys got pedigree. The reason why they're at almost a $2 billion market cap and we're at a $250 million market cap is because we were a bulletin board a month ago, and they went out with Goldman Sachs, and I had to go through the bulletin board route with debt. I had to clean up my balance sheet, and I just now got on stage. But I waited to take the company public until I had data that I could have the fist fight with. I started the company. I'm the founder. And then built it up, got it to a market cap of around 230 million. And then they decided they wanted to bring institutional management in. And I went, okay, I don't let my ego get in the way of what's right. And that was in 2018. And they didn't execute. They didn't get the trial into the clinical thing. And then kind of like Godfather Part 3, I thought I was out and they keep pulling me back in. They went, Tony, you got to get back in there and take this thing over. This management doesn't know what they're doing. And I'm like, this is what you guys wanted. And so I fired the entire board in 2019, got the trike back. I got it into clinic. Everything I said I was going to do, I did. And, and that's the roller coaster ride that we had, me personally. And everybody's happy now because we're performing. Execution, look, I hear many excuses for failure. I never heard one excuse for success, right? There are no excuses. You got to execute. You have to do the work. You have to have things that, and the reason I focus so much on data is because it's irrefutable. It is real science, real clinical data from the FDA. You can't lie about your data. That's how you go to jail. Data has to be exactly what it's articulated to be. Once you have data, then I knew we were ready for NASDAQ, and that's why I waited. So when someone said, well, you got a $25 price target from Roth, I went, yeah, that's really very low. Because even at $25, our market cap would be 600 million. We'd still be the low guy in the totem pole. Our data is outperforming everybody. Everybody has CRS, cytokine release syndrome, toxicity, except the trike. Everybody has issues and uses combination drug therapies because they need assistance, not the trike. So our data is showing things in phase 1 that nobody sees. It's very rare to see the phase 1 data like this. My first biotech company was Senatech PLC in 1994, I think it was. It's a while ago. That was my first biotech company. Then I went to GT Biopharma, HIV testing. That's how I know a lot about HIV, by the way. I left because I wanted to do something else at that time, and it performed really well. And then I created Iovance, right? From nothing to, it got as high as 7 billion. It's sitting at about 5 billion right now because I knew cell therapy was around the corner. So I'm either really damn lucky or I might actually know what I'm doing. I'm okay with either one of those, by the way. I don't care if it's luck or I actually understand what I'm doing. But I just think execution is really important and not focused enough. Nobody can do what we're doing because we have when I get CEOs that come up to me, because I went from idiot to genius overnight, I have pedigree now, right? They go, well, what's really important, Tony? And I go, you know what? You got to have data, but you have to have a really solid IP strategy. I don't like holes in patents, right? I tell them, don't go to your lowest bidding attorney. Spend money there. $1,000 an hour for DLA Piper, Dr. Lisa Hale, #1 patent attorney in the country. I spent $600,000 before I even licensed the trike. I don't like holes because ultimately, when you're in this game and you're showing data and you're showing commercial viability, Big Pharma is going to come in to partner with you or buy you. When they do, they have a team of 80 people, patent experts. If you have a hole in your patent, they will find it. I don't like holes. So you make sure you build that picket fence around your patents. You make sure it's solid. So that's, just you got to pay attention to the business model along with the technology model and the commercial model. It all has to be integrated. So there's a couple of things to that model, right? First of all, there's the endpoint cost of the technology, right? At, you know what builds your business model? It's called an FDA label. The approval label that you have on your drug tells you who you can sell it to, what they have to be to qualify to sell it to, under what conditions you can sell it to. So that's the first thing. You backfill from there. The other thing is manufacturing issues, right? You have to get a hold of that. The reason I didn't really want to do another cell therapy company is because the clinical trial cost alone is $300,000 to $500,000 per patient. My clinical trial cost with the trike is $30,000, and 70% of it is covered by insurance because of our use of IL-15. All that they pay for all the hospitalization. We don't really need to deliver the trike in the hospital, but they wanted the first phase 1 portion so that because this is a first in class, first that time ever this biology has been used. The trike, so it costs $2 million for the trike to set it up for manufacturing, to develop the paste, which is E. coli, CHO, mammalian, it's those kind of things. Once you do that, the manufacturing cost per trike is about $1,200 to $1,500. You can sell that trike for $180,000 to $250,000. Everybody else is more than 2 to 10 times more expensive. So the margins are 98%. And that is a model that I like economically. Okay, so here's the good thing about the FDA. When I heard a question before, it sounds too good to be true. There's no such thing in FDA data. If it's too good to be true, they close down your trial. There is no ambiguity into whether a technology works or not in an FDA trial. I have some people go, well, what about, you're treating patients, the placebo effect. There's no placebos in cancer trials, right? This isn't a blind study. Every patient you deal with is terminally ill. The FDA is not that cruel. The FDA requires that every patient get the drug. So that's what I meant. There's no ambiguity about the data that you have in this realm. 3 out of the last 5 patients have gotten CRs and or right. That's big. That's phase 2 approvable data in phase 1. All right. But everybody's different, right? So there will be cases where we will ultimately get a side effect. All right. This is amazing that we haven't yet. We will. Um, but if it does happen, it's probably very manageable because the safety of the trike, it's really easily tolerated. These patients haven't experienced this kind of thing in years because they've been through torture getting to this point to where they're trying an experimental drug. And then all of a sudden ours has no fevers or any of that stuff. That being said, some patients will get cured. Some will get survivability for 8 months or 3 months, and they'll need another realm. Every person's biology is different. So, of course, there'll be retreatments in this, which makes for a bigger economic model as well. Yeah, because if their NK cell levels, and by measurement, right, their NK cell levels, they're cured, you still want to keep those levels higher, and the trike has the ability to keep those NK cell levels persistent and continuing. So yeah, there'll be some, which is likely, by the way. So yeah, it is AML. One of the questions I had recently was, why did you pick AML? That's the toughest disease to handle. And I went, that's exactly why I picked it. Because if the trike can do what it's doing in the toughest disease, that's a multi-billion-dollar disease, right? For any therapeutic, multi-billions. Ibrutinib, $3 billion a year. It's an antibody. It's got cytokine release storm. It has a lot of blintumomab. Ibrutinib is a chemotherapy. Blintumomab is an antibody therapy. And they go through that same strategy. There's multiple billions. So that gives you valuation. So yeah, there is a strategy where the trike will be used. But we also have 22 other trikes. So we're going to continue to matriculate these assets through the clinical trials. It's extremely rare for phase 1 data like this. And in the cases where this has happened, every one of them were approved. So there is no case where phase 1 data was really stellar good and then phase 2. Phase 1 is all about establishing one thing, two major things: maximum tolerated dose before it causes problems, and safety profile. You get through that, the rest is all up to survivability and efficacy. We're already showing that in phase 1. There is in my opinion, this has been de-risked, right? I can't see, that doesn't mean something can't happen, right? But it's really rare for this kind of phase 1 data to happen, and it's extremely rare for it not to be approved. Yeah, those are two solid tumor trikes two infectious disease trikes. We're really good in oncology, but I don't think we understand the infectious disease world. I'm out licensing that to big pharma. They'll take it, and I'm happy with that. Let them handle it. We know what we do. We know what we know. We know what we don't know.
Right.
So that aspect of it, yeah, that's the benefit of the platform technology. It's really versatile. First milestone we hit by uplisting to NASDAQ on February 11th, right? Then we released interim data at the ASH conference, which made everybody go nuts and made the competition kind of start going, holy crap, these guys got something. All of the NK engagers, they're bispecific. They don't have IL-15. They all have partnerships, like I said, from Dragonfly to Affimed. And then we'll release data on patient 10 in about a month. We'll release data on patients 11 and 12 by the end of May, and we'll be starting phase 2 in June. Our solid tumor trikes start in for their enabling studies to get into clinical trials with the FDA for PD-L1, B7-H3, colorectal cancer, ovarian cancer, prostate cancer, breast cancer, triple negative breast cancer. We've got a lot of firepower.